Okay, good, because they're on our wristbands. Yeah. All righty. So with that, I would like to start by introducing Natalia and Miguel, who is going to introduce Patricia. So Natalia, the floor is.
Well, hi everyone, it's me again. It's a real pleasure for me to be here to introduce Patricia. I've been a postdoc in her lab like 15 years ago. And honestly, I feel really honored when they invited me to make this introduction, but I had no idea how a big deal the introduction is. So I'm a little bit nervous because I know that you are expecting jokes, videos, and I'm not gonna do that. It's not my language. I have limitations, so I did my best, but there's not gonna be like Boris did, because I look at all the introductions and that's not gonna be like this. I'm going to start talking about Patricia's beginning in her career. She earned a bachelor degree in biology in the Murray State University in 1978, and then she got a PhD in cellular and molecular biology at the University of Michigan. And before being the Trichomonas expert, as you all probably know her, she has been working in Amsterdam in a postdoctoral training at the lab of Pierre's board, working on antigenic variation. So she was previously a trypanosoma girl, and I was surprising to see that in her postdoc, she published 4 Cell papers. I cannot even publish one. And she published 4 Cell papers, and 4 of them were in the same year. And that talk a lot about how Patricia is. So she's a hardworking person, very smart, very dedicated, and for me, that's amazing. So after her postdoctoral work, she moved to UCLA, where she opened her lab as an assistant professor. And here is Patricia in her first office, and with her first PhD student. And as you can see, there is a traitor here, because Peter Bradley, before being a toxoplasma expert, he was a Trichomonas boy. And after she started her lab, she dedicated more than 3 decades to research on Trichomonas. And in 2019, she got promoted to the most senior level professor in UCLA. So she was distinguished professor, which is the highest rank in UC system. And all of this, she did it while being a great mom, a wife, and building a beautiful family. So she did it all, actually. And she received many recognitions and awards: the NIH Merit Award, the Alice and Cici Wang Award from ASBMB. She was elected to American Academy of Microbiology. She got the New Investigator and the Scholar Award from Burroughs Wellcome Fund. And finally, in 2019, she was selected as a member of the National Academy of Science. Probably that's the dream for many of us, but only a small portion of scientists can get that award. And fewer parasitologists. There are not that many parasitologists that can be member of the National Academy of Science. So I think we all agree that this is a very difficult achievement for any of us. Can you imagine how difficult it is if you work in a parasite that nobody cares, that only very few labs work? Well, I care. I care about it, but we just 3 people. So nobody cares. There is no tools, very few labs. So for me, this is actually amazing. I don't know how she was recognized by the National Academy of Science working in trichomonas. So that's Patricia. And as I said, she dedicated 3 decades to work on trichomonas. But some of us generally work in a very specific aspect of the biology of our parasite, but she worked in complete different things. She worked in gene transcription. She identified core promoter elements, transcription factors that allows us to understand a little bit of transcription in trichomonas. Then probably most of you know that trichomonas doesn't have a mitochondria. Instead, it has an organelle producing energy called a heterogenosoma. And she has been working for many years trying to understand the evolution and function of a heterogenosoma. She also worked in drought resistant. The treatment of trichomonas is based on metronidazole, and there are many resistant strains. So she has been trying to understand the mechanism of resistance to metronidazole. She also worked in host-parasite interaction, identifying the lipoglycan and surface proteins that are involved in the interaction with the host cells. And during the last years, she has been working in extracellular vesicles as a mechanism of communication between the parasite and the host cells, as well as the parasite communication. So as you can see, a very diverse area of study. But I don't want to spend much time talking about her science because that's something that she will do. I'd rather spend time talking about something that you probably don't realize about Patricia. And that's... No, don't be scared. Don't be scared, no. So her science was not selfish because she always knew that she was the biggest lab in the field with more money, with more people. And I remember her saying that she always needs to put some effort and money for tools development because if she doesn't do that, nobody will. And she was right because we don't have that many molecular tools, but the ones that we have are coming from Patricia's lab. And she developed the transfection protocols that allowed us to overexpress genes in trichomonas. She also developed a tetracycline inducible system. She characterized 26 different strains in pathogenesis-related phenotypes that allows us to study pathogenesis in different labs of the world. And she tried very, very hard to develop knockouts at the beginning by homologous recombination and then by CRISPR-Cas9. And although it's doable because she published it, it doesn't work that well. And finally, she also co-led, along with Shane Carton, the first genome project that we have. And Shane sent a message that I like to share with you because it talks about the way that Patricia analyzed the science. In 2002, Patricia convinced Shane Carton to start to sequence trichomonas for the first time because Shane has a lot of experience in genomics in Plasmodium. So they started to work together, but they believed that the genome was going to be like 15 or 20 megabase, and it was not like that. Trichomonas has a very complicated genome, 160 megabase, very highly repetitive, complicated. So when Shane started sequencing the genomes and the sequencing keep going and going well past the 16 megabase, she said that Patricia didn't lose trust on her and she didn't think that she was sequencing the wrong organism. And that's the way that Patricia analyzed the science. She let the science speak for herself. I think that's a lot, and that's the way that we all should work. And after, I also asked for messages for other people because although I am the one that is standing up here, she was important for many of us. And I was surprised to see that many people highlight the role model that Patricia was for women in science. One of her postdocs, Katie, she said that she meant a lot because she went from the only woman in the department to the most senior level professor working on the UC system, leading a lab filled of women, focusing on women's health. And then when I started receiving messages, I always felt that Patricia was my role model. So it's the type of science that I would like to be. And when I received messages from 2 other PIs, one of them that is here, Angelica, and Francie, they both highlight that Patricia, she was the role model for them being the PI that they are today. And Angelica sent this message that I think is very strong, and I hope I could be so influential in the life of somebody. And she said, Patricia is the reason that I became parasitologist. After taking her parasitology class, I fell in love with parasites, then performing research in her lab made me fall in love with trichomonas, and she's my strongest female PI role model. So that's a lot. No? Being a lot of people that you inspire, I don't think it's something common. And she mentored a lot of people. She was a very good leader. Her lab, it was always a nice place to be. It was always very friendly, very collaborative. And that's not a coincidence if it happens along more than 30 years. It means that you know how to be a good leader, and it means how to manage groups. And that's something that Patricia do very well, because along all this year, it was a wonderful place to be. I think it was my best decision has been made a postdoc in Patricia's lab in my career, right? In my life. I have a husband too, no? And I think I know the secret, because all the things in Patricia's lab happen around food. There is always an excuse to have a cake. If there is a birthday, you have a celebration and you have lunch. And maybe she didn't say that, but maybe that's her secret. And I think it's good. And as I said, we are a very small community. There are not that many labs working in trichomonas, but most of the PI that we are leading labs in trichomonas are coming from Patricia's lab. So 5 labs around in US, 1 in Argentina, and 1 in New Zealand. All of us are coming from Patricia's lab. And this is the message I received from the newest PI that came from Patricia's lab. He's Josh, and I like the message because if you know Patricia, it's this message. So Patricia can talk your ears off. I go to ask her for help on an idea, on an experiment, and she will have multiple suggestions, and then she'll be talking about to me for another hour about my daughter's birthday and anything else that is going on in my life. And that, if you talk to Patricia, that's the way she is. And probably that's her secret to have a successful lab. She cares about science, but she also cares about people. And that means a lot for everybody that is working in the lab. And she mentored not only students, and she helped not only her own students. She also is a scientific grandma, because in my personal case, I have a lab in Argentina, and she always knew that for me it's very difficult to make good science living in a Latin American country. So since I came back to Argentina, she always helped me trying to have international grants, or each time that I need to make an experiment that I cannot do in Argentina because I don't have the equipment, she received my students at UCLA and she mentored them. So for me, it means a lot because she's not only my role model, so she's also my best collaborator. So I think it's clear that she was very important for the trichomonas community. But not only for the Trichomonas community, she was important for the parasitology community as a whole, because she was an editor for many parasitology journals, such as PLOS Pathogens, MBio, MVP, Sphere, Experimental Parasitology. She was a member of the study section of NIH for many years. She organized international conferences, and she was a professor from Bob since 1995. So more than 30 years being a member of this community and being a professor in Woods Hole. So, and this is a picture that Peter sent me, and he said that they used to put money on this Buddha so to have the experiment work, and then somebody steal the money, and they put the money again. But he also said that he was the one that always put the money. I don't know if that's true, but he said that. He said that. So, and she was so supportive of the Bob course that she also was the director of the course from 2004 to 2008. Probably some of you are also on this picture, but she was very supportive of the Woods Hole course as well. And I think the most important part of this talk is the person behind the science. So Patricia is a person that she cares about people, and she always tries to help the people that is in the lab and the family. If you go, the family is always welcome in Patricia's lab. You can see kids around in the lab when we were a postdoc there. So so she's a person that listened, she encouraged, and she always tried to help. And this is something important. And I would like to finish with this picture of myself with my baby. And this is and probably you ask why. And the reason is the rocking chair. Because when I was pregnant in her lab, she offered me the rocking chair that she used to carry her babies. And at the beginning, I didn't want to because I have a small apartment and everybody said, yes, you have to take the rocking chair. So I took it, and then I like to think that we both carry our babies in the same rocking chair. And I love it so much that when I came back to Argentina, I took the rocking chair with me because it was one of my favorite places. And now the rocking chair is the favorite place of my kid. So thank you, Patricia, for the rocking chair. And I would like to end with a message that Peter sent me. And I think it's very nice because, especially in this moment, because Patricia, she always tells him that we are so lucky to be able to do what we do. And this talks a lot about her passion for the field. So I think it was a nice sentence to close the presentation. And thank you, Patricia, for your work in Trichomonas and for changing many careers, including mine. Thank you.
Thank you very much. Thank you, Natalia. Be rest assured, we're really looking forward to hearing your talk, Patricia, but we're going to, if you wouldn't mind, we're going to delay just for a couple more minutes, maybe extend your discomfort for a couple more minutes. So within our community, we've got a young man with a rare combination of talents. He's not only a very fine young scientist who chaired one of the sessions earlier on, but he's an excellent artist as well. And he spent a little bit of time preparing a gift for you to commemorate your achievements over the year. He's very shy, but we've convinced him, rather than me giving you the gift, Nathan Klassen is going to give you the gift himself.
Oh, thank you so much. It's so special.
So for those of you that you can't quite see it, it's up on the screen. So talk later, yeah.
Very special. Thank you so much.
I can assure you, you're now allowed to present your talk if you
got
want.
the anatomy really close. There's an axis style. Thank you so much.
Over to Patricia. Thank you very much.
Well, everybody says I talk so much, but I'm suddenly speechless. Anyway, I so appreciate your introduction and all your kind words. Honestly, oh boy, I'm losing my voice now. Let's try this. As Peter said, we're all so lucky. We're so privileged to be able to do the things we do and to enjoy our science, even get paid. Maybe not that much, but to get paid. You can at least buy a house. Well, young scientists, maybe not, but us old guys who started before the prices went way too high. But anyway, it's just been a privilege. It's been an incredible privilege for me to not just be able to do my science, but to have all of you in my lab and the people you mentioned, and have the privilege of being your mentor and helping you out and you helping me out. I always say when I have to give talks like this, or I'm getting an award or something, you forgot to say I got the best mentoring award, postdoc mentoring award from UCLA. Not that I'm modest, but you know, I always say, like I said, when I had to respond to that, when they gave me the plaque, is I've learned as much or more from everyone who's worked with me as they have from me. So it's it goes both ways, and the pleasure is an honor has been mine. But before I say more, thank you, Natalia, and thank you, Angelica, thank you, everyone. I want to thank the organizers for giving me this opportunity, for inviting me to give this keynote. I think Jane gave it away in her congratulations on your retirement. I retired last year. And so wasn't really necessarily expecting that I would give another talk. So I just really am honored. I'm honored that you asked me. And this place is a very special place to me. As Natalia has said, I've spent many weeks here. In fact, I think at one time I calculated it, and if you add them all up over the many years I've been here, probably been here 8 months out of my life or something, but because of the Bob course. And so it's a very special place to come to. It's a very special for me to be here. And I also want to thank the organizers for having organized such a successful meeting. You know, we all know this is not so easy. And so could we all give a round of applause? Okay, let's see if I can figure out how to work this pointer. Omar, why does, okay. Yeah, it's this one, right? Can you guys see it? It's like flipping around here. Okay. So today I'm going to talk about 2 projects in the lab. My lab being, as you know, from the introduction at UCLA. And the first one, and they both hopefully give some cellular perspective. Oh, no wonder. How many times did you tell me to get the right one? I know. Okay. Okay. This is, I know, sorry, it's not boding well, is it, huh? Okay. The first one I'm going to talk, first project, which will be about 2/3 of the way through the talk. And if it's gone so long at that point, you guys can all just get up, leave, and that'll be the way to tell me you've had enough. This one is about secreted extracellular vesicles secreted by the parasite and their impact on infection. And the second project is going to be how, and it's shorter, what I tell you is much shorter, is going to be how neutrophils that are shown in magenta here surrounding the parasite, shown in green, of how they killed Trichomonas. But first, I'd like to give a short ancestral history of trichomonads. This parasite is an excavata. I'm sorry, but did I do something? Okay, I obviously did. It's in the supergroup excavata, and you see that supergroup here. What you're seeing here is a tree. The colored lineages are eukaryotes. They're all arriving from the archaea here. And this is the excavata and the yellow, orange, and purplish colors. And it's in the phylum Parabasalia right here. And you see its nearest neighbor, this is a parasitology meeting, so I'll put some names on here. Its nearest evolutionary cousin is Giardia. You have the kinetoplast here. Up in this area, these lineages, Antamoeba, and then Toxoplasma Plasmodium cryptosporidium. But the point I want to make is that a lot of work has been done by many very, very good evolutionary biologists over many years, and using different gene sets, using different methods, lots of different methods, and these days, lots of different gene sets, trying to determine what is the last common eukaryotic, last eukaryotic common ancestor. And everyone is, I think, pretty much agreeing now that it's the Parabasalia. And so this parasite, it comes from this lineage that really is giving you an insight into very early evolution of eukaryotes. This is Sandy Baldolph's tree. I should have said that. But Andrew Roger published something using different data sets but different methods and got the same thing not that long ago as well. Oops, sorry, wrong way. Okay, here's my graph of the parasite. So you can see some really nice cellular features here. Not very, very high resolution, but they still stand out. 4 anterior flagella, the 5th undulating membrane, the flagella that's connected to the membrane. So these Parabasalia are characterized always by multiple flagella, and they have a very complex cytoskeletal system that is often flagello associated. They have the parabasal filaments, and these parabasal filaments actually connect the basal bodies at the base of the flagella to the Golgi and sort of cage the Golgi. They have a costa, which is actually supporting this undulating membrane. They have an axostyle, which is this protrusion here. And it actually starts here. It basically runs the whole area of the body from the anterior end under the nucleus, as you see here, and protrudes at the bottom of the cell. There's other features of the parasite that are very unusual, and reference was just made to this. They lack aerobic mitochondria, but instead have an anaerobic hydrogenosome, or in some Parabasalia, they're mitosomes that have a common ancestor with the mitochondria. They came from the same common alpha proteobacteria endosymbiont. Now, I spent, as now Natalia has indicated, I spent a good portion of my career, probably half of it, more than half of it, studying some of these really interesting cellular features, particularly the hydrogenosome of the parasite, because I just thought the biology, and the cell biology was so fascinating. And then in the last part of my career, I have turned then more to post-pathogen interactions, but I've actually come full circle now. I'm not going to tell you about the data today, but I'm so happy after years of trying to get my colleague Hong Zhou at UCLA, who's a cryo-EM expert, to collaborate with me. He's been collaborating on things that I didn't really want to do, but he's finally getting the structures we're getting the structures of the parabasal filament and the costa And so I kind of like the idea that I start off looking at really basic biology and unusual features of this organism, and then maybe the last few papers that I'll publish will be on something, and the data we have it's quite interesting. It's different. Okay, key milestones in the study of T. vaginalis, and then I'll move on to talking about my own research. In 1836, the parasite was discovered by a French public health doctor, Alfred Donnet, and he named it Trichomonas vaginalis. And he named it that, tricho, hair, I've been told in Greece, monas is a single body, and vaginalis because he was observing it in the vaginal discharge of Parisian prostitutes. In 1916, Hohn, a German physician, recognized that the parasite caused a clinical condition, and that condition is referred to as trichomoniasis. In the mid-20th century, trichomonas was recognized as a sexually transmitted disease, not just an infection, but sexually transmitted. In 1973, B.M. Holmberg reported the first DNA isolations. 1975, Miklos Müller and his colleagues identified and isolated these very interesting and important organelles, the hydrogenosome. And so those of us in the field sort of feel that Honenberg and Miklos Müller are really the founding fathers of modern day trichomonas research. In the 90s, the first gene was cloned, molecular biological techniques were first applied. 1996 was the first evidence of the hydrogenosome and mitochondria evolving from the same common endosymbiont. And then, as Natalia's already mentioned, in 2007, the first genome sequence of Trichomonas, which was spearheaded by Jane Carlton, was published, which of course has paved the way for much more detailed work, as well as better clinical studies. And Natalia's already told you a bit about this genome. It was surprising. It is very large. One of the reasons it's so large is about 2/3 of the genome is repetitive. There's a lot of transposable elements amongst those repetitive elements, but there's also a lot of really, really high copy number gene families. So we have GTPase the small GTPases. I think there's only 400 of them. I mean, we have kinases. It's crazy how many very large gene families we have. And there's very good evidence of lateral gene transfer. And so a lot of these genes, it looked at, or not a lot, but there are many examples of transfer from bacteria, usually anaerobic bacteria that live in the gut. And then now from 2010 onward, there's been identification of virulence factors, studies on the two common endosymbionts, the double-strand RNA virus that was talked about by Wynn 2 days ago, and a little teaser talk up here, as well as a bacterial endosymbiont of Mycoplasma hominis. And even more recently, people have started studying, as one person being Angelica, the cervical vaginal microbiome and how that infects infection. So that's sort of my short history on things that people usually don't talk about when they don't have very much time. You know, of course, that it's sexually transmitted. T. vaginalis is strictly a human parasite. There's other trichomonads that infect all sorts of different mammals and even lizards and so forth. The one that's best studied after T. vaginalis would be Trichomonas foetus, which is studied in cattle. Or it's not studied in cattle. Well, it is, but rarely. But it infects cattle. It's microaerophilic. I really apologize. Do you think I'm doing something? Well, I guess I am, but I don't know what it is. I'll try not to punch anything but that. It's microaerophilic, lumen dwelling, okay? It resides in the urogenital tract, not the least bit not the least bit surprising. There's about 250 million cases of trichomoniasis worldwide, with greater than 3 million right here in the United States. and published from on the CDC website just this year, a survey showed that over 2% of US women of reproductive age are positive for T. vaginalis. So it's quite common, as you see. It's basically a neglected infection of poverty. If you look in intercities and with certain racial groups, African American, you'll find 10% infection rates as opposed to 2% overall. It has been classified as a neglected parasitic disease by the CDC about 10 years ago now. Both men and women are infected, but it's a clever parasite in that most men are up to 95% are asymptomatic. And why do I say that's clever? They go about spreading it, not even knowing they have it. Whereas with women, the symptoms, about 50% are symptomatic. The studies, the clinical studies, the epidemiology studies sort of, they vary in the number they give, but on average, mostly they come out to about 50% have symptoms. If you look at this, this pyramid here, that you'll see that if you add up syphilis, gonorrhea, and chlamydia infections, this is US data, that all of those three bacterial STIs are less common than just trichomonas alone. And this is just another to drive that home, that here's trichomonas in terms of prevalence, and in the United States, only human papillomavirus and herpes simplex virus are more prevalent. Natalia went through this this morning. Most of you perhaps were there. I'll go really quickly. Its infections can cause vaginitis, urethritis, procystitis, and so basically a lot of inflammation. And it increases the risk of HIV transmission. And you see here, this is an infected cervix. So the parasite resides in the vaginal tract in women, but it really likes the cervix. That's where it really hones in. And this is an infected cervix. And they call this a strawberry cervix. And the reason they call it that, you see all these darker spots here? They look like the seeds on the outside of a strawberry. These are lesions. So this parasite can be very lytic and cause a lot of tissue damage. And we think that the reason there's an increased risk of HIV transmission when you have this parasite is it's just a portal of entry for the virus to go in. Long-term infections, if they're not cleared, have severe consequences such as increased risk of preterm birth, infant mortality, and urogenital cancers, and even infertility. Okay, I want to make it really emphasize that it's an extracellular parasite, because so many of the talks and so many of the people in the room, of course, work with a parasite that dives right into the cell, and everything goes on when the parasite's actually inside the host cell. But this stays strictly extracellular. But it must adhere to the host cell to both establish and maintain the infection. The parasites, once they're adhered, they get a lot of essential nutrients that they can't make themselves, that they have to obtain from the host by host cell lysis and then phagocytosis of the material. And you saw a nice example of the really serious host cell lysis on that last picture. This lysis is strictly dependent on adherence. So adherence is really key for this parasite to be able to establish infection. There are a reasonable number of factors, but still that are involved in this adherence in lysis, but they're still quite poorly defined. There's an abundant lipoglycan that we know is mediating parasite-host interactions. It binds with a host a receptor called galectin. There's extracellular cysteine proteases. There's many parasite surface proteins. So as the years have gone by, my lab, but a number of other labs, and Natalia and Angelica here have certainly contributed this, have shown pretty strong evidence, even as much as gene knockout evidence of so many different parasite surface proteins that are involved in binding hosts. So it's very multifactorial. There's not, like one key, one lock. There's many keys, many locks. And there are also secreted proteins that have been shown to be involved in establishing this infection. What I'm going to talk about right now, though, is whether or not I'll show you the evidence that secreted vesicles, and specifically extracellular vesicles that are referred to as EVs, or exosomes, that they play a role in establishing the phenotype. What are extracellular vesicles? They're very small vesicles. They usually range from 30 to 120 nanometer in diameter. And you'll see here, they get made within a cell in a late endosomal compartment, a specific type of a late endosome called the multivesicular body. So these little green vesicles are what we're talking about. And then that multivesicular body goes to the plasma membrane, fuses with the plasma membrane, and secretes out the exosomes. So what do they do? Why is this happening? Well, these exosomes were actually recognized many years ago, decades ago actually, in mammalian cells, but they were sort of cast aside as garbage, blebbed off garbage for many, many years. And even today, the vast majority of the work is still done in the mammalian cell systems. And in those systems, they have been shown to play a role in cancer, immunology, as well as neurobiology. And how this is happening is the exosome, as shown in this cartoon. There's a donor cell that secretes out the exosome, and then it is internalized by an acceptor cell. And so they're great ways by which you can take cargo from one cell type to the other. So they're involved in intercellular communication. And this cargo, I'll draw a cartoon here, it's protein, it's RNA, it's lipids, it's pretty much anything that you would normally have as building blocks in a cell per se. Now, when we first started working on EVs, there were 2 or 3 other parasite labs that all started at the same time, mine being one of them. We actually didn't know that we were working we didn't know about each other's work at the time. It really wasn't thought that exosomes were that widely spread evolutionarily, but they're now known to be produced by all living cells, okay? Even bacteria produces them. In the bacteria field, they call them outer membrane vesicles, but they're exosomes, and they agree with that. Protists, of course. I think some talks here have already talked about exosomes from their parasite. I think all the parasites that have been examined so far have been found now to have them. Fungi have them, and of course, multicellular eukaryotes. And in all these different systems, they don't do the same thing. One thing they have in common is they're transferring information from one cell to the other, but they're involved in a vast array of biological processes. And there's an emerging field to use these as biomarkers of disease, and less developed is to deliver them deliver drugs or vaccines. But one of the stories that I've followed kind of closely, because, well, you don't need to know why, but I have, that has nothing to do with parasitology, is exosomes that come from a diseased Parkinson brain have different markers on them. There's 2 or 3, and there's even an RNA that are not coming from the same brain tissue before it becomes diseased in Parkinson's. And as a result, they have now developed ways to diagnose Parkinson's earlier, because Parkinson, way earlier than any symptoms. And previously, the only time they even could say possibly someone had the disease was when they were already exhibiting symptoms. So I think we'll hear more of that, maybe in parasite systems, but certainly in mammalian systems. Okay, so this study, the exosome, we started working on exosome because of Natalia. She always, I always did what she wanted me to do. I let her do what she wanted to do. I always did what she wanted me to do. But she made a very interesting observation, working on a totally different project. You may remember, I'll try not to sidetrack too much. You may remember you came in and showed it to me. I bet you don't remember this. And I said, it's an artifact. And then I said, go redo it. Go redo the experiment. Let's see. If you can reproduce it a few times, maybe you got something here. But it made her think, maybe these parasites were making exosomes. And she was right. And she joined up with Olivia too, an MD PhD student in the lab. And the two of them did a number of the initial experiments. I'm not going to show those. That showed that the parasite does make exosomes. Actually, I am showing one or two, sorry. Does make exosomes and delivers the exosomes to the host cell. And these are those experiments. They modified pre-existing protocols that were being used to isolate mammalian and human. I'm doing it again. There's something, I'm so sorry, guys. There's something, some way when I use the pointer, I also turn something off. They modified protocols that maybe I won't point. You know what I'll do? I won't point unless I really have to. Because I seem to be very uncoordinated with this pointer. The human, you see it there, I should give them credit where I got that. That's they modified protocols that were allowing the mammalian people to isolate these exosomes. And sure enough, when we did the staining and looking under the microscope, the EM, at what came out of their EV preps, their trichomonas preps, they look very much like oh, okay. This is rather embarrassing, isn't it? Omar. I'll go on, but I'm just not understand. I'm just hitting what I'm supposed to hit. And you think I'm hitting this thing? Okay. I will try not. Okay, they look very much like had been published for human exosomes. It turns out later in time that we found out this cup-shaped thing is an artifact of this particular method for isolation, but never mind. And then they looked at the size of the particles, and in fact, that's showing here with the nanosite machine, that's showing you this here. I'm really sorry. I'm not going to point. I said that before, didn't I? Oh, I can walk. I can point with my finger. Oh, it's not gonna work either? Oh, come on, I shout. Okay, now then I'm trying to do 2 minutes. I can't coordinate all this. Okay, will that work? Woohoo! Let's try it. Okay, but right now I don't want to use the space bar. So you see with this nanosite, yeah, so I woke David Roos up. Even David's laughing now. This nanosite that reads millions of particles shows the mean diameter of them being more or less 95. I told you they're usually, sorry, they're usually 30 to 120 nanometers in diameter. And the thing that really sold us on the fact that we were looking at exosomes was they did a proteome. And this was quite a long time ago, so we didn't at that time get nearly as many proteins as we get from it now. We now get twice as many. The technology mass spec technology has gotten so much better. But it starred here in red are proteins that in that proteome were known to be really conserved proteins in mammalian exosomes. And everyone in this room doesn't have to be told how, and this is just like by blast analysis. It don't need to be told, you know, how divergent so many of our proteins are in our genomes, and particularly Trichomonas is that way. And so at that point, we felt we definitely were working with, I can use this, great. We were working with exosomes, and we had shown that they were delivered to the host cell. That was one of our very first paper. And then a postdoc in the lab, Anand Ray, asked how are these parasite EVs delivered and taken up by the host? And he just basically took the EVs, labeled them up with CFSE to make them fluorescently green, and then put them together with the host cells and used flow cytometry to see if they were getting taken up. And this is just his establishing the assay. Yes, they're getting taken up, time dependent, temperature dependent, only get taken up at 37 degrees instead of 4 degrees, showing that it's an active process. He was looking at the proteome that had been derived, and one of the reasons we were scared were looking specifically was for maybe identifying the ligand of the EVs that bind to the host cell. And it sure did provide some clues. And what he found is that one of the very abundant membrane proteins is predicted to bind heteropolysaccharides. And those of you who work with eukaryotic cells already know where this is going. So are there host cell proteins that are containing the heteropolysaccharides? It's specifically some of his structural analysis prior to this type of thing was saying they thought it might even bind heparan sulfate. So where do you find these things? You find these things in the glycoaminoglycans that are on the surface of many host cells, cartooned here. These gags are repeating disaccharides. They're linear disaccharides. They can be attached to proteins, usually are, or sometimes attached to lipid. In the cell types that we were looking at, the epithelial cells from the urogenital tract, the most abundant gags are heparan sulfate and chondroitin sulfate. And so I contacted Jeff Esco down at UCSD. Jeff, as some of you may know, is the guru on anything glycosylated or anything glycan related. And he had made Chinese hamster ovary mutants, one of which does not make any heparan sulfate, one of which makes no gags at all. And so he gave us those cells, and this just shows you the data from that. I'm going to try this again. I guess I'm a glutton for punishment, or for punishing you. This shows that when Anand did the uptake assays with, no, I am a glutton for punishment. When Anand does the uptake assays with the wild type CHO, this and sets it to 100%, that's the uptake. But if you knock it down quite substantially, just if you have used the mutant that doesn't have heparan sulfate, and even more so with GAGs. Now you're going to notice this immediately and say, but you don't really knock it down all the way. And this is true. But it's clearly playing a role. And then we did this by the 3 different ways, partly because we weren't getting absolute a complete knockdown. But if you take heparan sulfate and do a competition reaction in the uptake assay, and that's what's being shown here, different concentrations of heparan sulfate, that also is knocking down uptake. And you can even remove heparan sulfate from the surface of the CHO cells using a specific enzyme, lyase, and that also will knock down the uptake. So then we thought, okay, well, this protein that is predicted to bind heparin sulfate. It looks like heparin sulfate is involved in an uptake of the parasite exosomes into the host cell. And so purified the protein, cloned it and overexpressed it in E. coli and purified it. And then he did an ITC analysis, as you see here, that's testing, is this protein binding specifically to heparin sulfate? It was done in such a way that you can even get the Km from the curve here, the Km for binding. It has a very tight binding, about 500 nanomolar, and then of course the control is nothing's happening. Then he competes with this protein. So we were saying, if this protein is really involved as the receptor and you put lots and lots of it in, it's going to bind up the sites and then it's going to stop the uptake of the EVs. And sure enough, that's what happened. This is just the regular assay, no extra protein added, just the exosomes and the host cells. You put in just good old BSA, nothing happens, but it really knocks down far with this protein. So through these studies, he was able to identify the EV ligand for the host cell uptake, as well as a putative host cell EV receptor. Okay. Okay, now, how does EV uptake affect parasite adherence? Because that's what we were really wanting to know, is it involved in that? And do EVs from highly adherent parasites have a greater effect on adherence? And to appreciate this, you need to realize that there's a really large range. This shows a 25-fold range in adherence of clinical isolates to host cells. Okay, so these wimpy parasites down here, that's showing you the percent attachment, very, very low, and this is to our two ectocervical and our prostate cell lines, our model cell lines. These are really very low adherence to both of those cell lines, and it increases toward the right side of the graph. And if you look at the difference between the lowest and the highest, you see about a 25-fold range. And these just happen to be 26 lines that we decided to test. I mean, I'm sure if we were doing a lot more, we'd probably even find a broader range. So what we did here, this was done by Olivia actually, when she was still in the lab. We took exosomes from 3 of these lowly adherent parasites, okay? And we took exosomes from a few of these, well, 3 of these more highly adherent parasites. And then we said, if we add those exosomes back to the host cell and we try to bind with this wimpy parasite called G3, it's the genome strain, what do we see? And this is when you're doing those experiments trying to get G3 to bind to host cells. If you use G3 exosomes, no good, nothing happens. T1, no. This one, these are all lowly adhering parasites. But if you use the more highly adhering parasites, as you can see, you get a lot more adherence, okay? And so this did establish that the highly adhering parasites are producing EVs that have something on their surface or I shouldn't say surface, we actually know that some of it is surface, but never mind. They have contents, their contents make them much more capable of mediating that host cell adherence. And so this was all in vitro. So Sandeep, that you can't see his name here, sorry, Mukherjee, was in the lab. This is later, after we had developed a mouse model for parasite survival, which depends on the parasite adhering to the host cells. And Sandeep took this model that we had, and he asked if I add the TV EVs in with the parasite inoculum, what happens? And what you can see here is that this is without EVs, this is with EVs, without EVs, with EVs. What it does, the EVs actually do increase the parasite load. And hence, we think evidence for consistent with it increasing the adherence of these parasites in an in vivo model in a mouse. So it validates the in vitro data that we've been getting up to this point. So then Josh Kochanowski, oops, sorry, Josh, if you're listening. Anyway, joined the lab. And I think he was one of the people that Natalia mentioned recently, or mentioned in her introduction. And so he did a nice assay that was more sophisticated than some of the stuff we'd done before, asking what the effect the EVs have on survival. And so he took the different host cells. This just happens to be one of them. He's done it with both the cervical and the prostate-derived cells. He took those cells at a 96-well plate. He either treated them with BSA or the EVs for 6 hours, then infected with Trichomonas. And then 24 hours later, he quantified TB, the parasite number, and as well the host cell survival in a high throughput assay using the operetta. And I should say that the EVs that we are using and have used, let me go back, I meant to say that and I didn't. They're from this strain, B7RC2, that really are very potent in increasing adherence. And it is one of the most adherent of the strains that we've looked at, and most lytic. It lyses the cells much more so too. And one of the reasons we're doing that is because of what I just said to you, but also we've screened, we were trying to do another project in the lab and we realized that it's very hard to separate the double-strand RNA virus from EVs when we do it's not a purification, that's the problem, when we do the isolation. And we tried very hard and it wasn't happening. And so we needed to find a strain that didn't have the virus, because there's all this murky, muddy, unclear literature out there that's claiming the virus does or does not have effects on adherence, this, that, and the other. So we just didn't want the virus anywhere near us. And this strain is the one that we're going to be using and have been using for these types of studies. It's completely lacking the double-strand virus. So what did Josh see? He showed, not surprisingly, that the number of TVs is increased considerably, as shown here, 24 hours post-infection, as you hear, and the number of host cells, conversely, is dropped down much lower. So in this MOI of infection and this 24-hour incubation, we usually have about 60% of surviving host cells. That's just the control. And it was knocked down at least half here. So then Josh asked, how does the parasite EV internalization affect the host cell transcription? So it's just basically an mRNA-seq analysis where you've got the EVs added for 6 hours, or a MOC, which is just a solution that's the EV's a vehicle basically, for 6 hours, then extracting the RNA and sending the stuff to be sequenced. And he showed that the uptake has a very large suppressive effect on the host cell gene expression. So in this heat map here, you can see this is suppression, purple, and you can see that the vast majority of the genes that were regulated were downregulated as opposed to upregulated. And then in doing this hallmark gene analysis that you see here, something really popped out that wasn't so surprising, because the parasites like to downregulate host immune response. But the most highly downregulated response was from the interferon alpha response. And we hadn't expected to see that. This is an extracellular parasite, and the interferon type 1 responses are usually associated with intracellular parasites or viruses. I want to first just tell you a little bit about, I just want to show you data that says this really is happening. So this is just a reminder of the type 1 interferon pathway. You often have, it's the ones that are talked about and studied the most are the alpha and the beta interferon. They bind the receptor, they set off a signaling cascade with JAK and STAT eventually gets phosphorylated, and after being phosphorylated, gets moved into the nucleus. And then once it gets into the nucleus, it obviously regulates a whole set of genes that are based on this pathway. What we found was that, or what Josh found, was there was no change at all in the interferon alpha or beta by the introducing EVs. But instead, there was a non-canonical interferon. It's called epsilon. You can see it here. It's quite highly downregulated. But also all the other players that were looked at in the type 1 interferon pathway were likewise strongly downregulated. Here's STAT1, STAT2, etc. So he got from David Sibley, who sent it 2 days before we asked for it. No, he sent it right away. He got a reporter just to double test that in fact the cells will be responsive to an interferon, and he cloned this reporter, which drives expression of luciferase. He cloned it into our cell lines, and then treated either with the EVs or the control MALK, 6 hours, then treated with interferon epsilon, and then 2 hours later looked to see, was he getting expression? MALK, obviously no. This is a fold change, so MALK is at 1. If you add just the TV EVs, no. But if you add interferon, yes, you do get. And if you add the TVs and the interferon, it now knocks this down. So the TVs are really knocking down this pathway. They're disrupting the signaling. He also went on, and I think I'll go faster here because I think I'm not getting down as fast as I want to. So I just want to say that he also showed that he was blocking the entrance, the phosphorylation and the entrance of STAT1 and STAT2 into the nucleus. So you just see there, MALK, it doesn't go in the nucleus. If you just treat with the interferon epsilon, yes, but then you block it if you have the TVs there. And that's all quantified here. In fact, I should have just shown you the quantification. It's blocked quite a lot when you have the EVs. So we were convinced at that point that this interferon epsilon was being downregulated by EVs. So what is interferon epsilon? I didn't know what it was. I had never heard of it when Josh showed me the data. And I was happy to hear that he hadn't either. And well, I don't know, should it have made me happy? I don't know. And so of course we had to go read. I mean, every time you find something that you don't expect, you go read. And there's a few papers out there on it, not a lot, quite honestly. But the one that really struck us is this paper that was published from a group in Melbourne about 10 years ago, actually. And what they show here is that this epsilon plays a key role in protection of the female urogenital tract from known sexually transmitted bacteria and viruses, okay? They also show that it's constitutively expressed in epithelial cells from the urogenital tract of men and women, okay? It's not it's not PRR stimulated, controlled by PRR stimulation. No toll-like receptors involved, no stimulation or regulation this way, but it's hormonally regulated by estrogen. Okay. And the levels fluctuate within the menstrual cycle. And what else was in the paper? Do I have that slide yet? No, I don't. I should have put it here. Anyway, they showed that interferon epsilon was much lower in peri and postmenopausal women. And when I saw this, I immediately said, wow, that is so cool, because there's been this literature out there for quite some time now that shows that peri and postmenopausal women have a much higher incidence of T. vaginalis than you see with any other sexually transmitted infection. Now, I'm showing you chlamydia here, but it's been shown for other things. I want you just to look at the positivity rate, the green balls here. And what you can see, it's not an unusual or even surprising finding, is that for most sexually transmitted diseases, you have a peak in positivity. Oh, I forgot to tell you this. This is so cool. They swabbed the vaginal tract of females from age 10 to age 80, and they collected more than 1 million. I'm not joking. I don't know what they were doing. And see, I'm trying to keep David awake. He laughed again. David, I'm working on it, David. But no, I mean, that's a lot of swabbing. Huh? And so it's impressive what they were able to do. But what the point is that you typically see the highest positivity rates in teenagers and, early, late 20s, so forth, and then it drops off. Well, with trichomonas, you see that too. And then you see a peak right around 50, which is some of us, about half of us in the room maybe, realize is perimenopausal or menopausal. And then we thought that this was really cool, that that there was a downregulation, and that led us to ask, could the declining, we haven't answered this question, don't get too excited, could the declining interferon epsilon levels during the peri and postmenopausal time contribute to the increase in the T. vaginalis infection? And I'm just showing you the data here. This is, I showed you a minute ago. This is the data from that paper that I should have showed you. This is how little is expressed at this time of the interferon epsilon. And so Josh went back and got no, I'm sorry, he hasn't done that yet, sorry. The first thing to ask, of course, is interferon epsilon protective against T. vaginalis infection? Because that would be the first step to say that, well, maybe this is why peri and postmenopausal women are getting infected so much more. So he goes and he basically takes the host cells, treats it with interferon for a while, infects with TV, and then quantitates the parasite load as well as the host cell survival. And let's just look for now at the quantitation. There's the pretty pictures that were quantified. But what you see is that it indeed is protective. Here's host cell survival. In the mock, under these infection conditions, we've only got about 30% of the cells surviving, but you pre-treat with this interferon epsilon, it's almost 100, it's really shocking. Same thing, the parasite levels drop if you treat with, so it does appear to be protective. Well, the other strange thing that Josh and I started shaking our head about and we did talk a lot, it's true, but a lot of science, but yeah, about his little girl too, but anyway, is why is an extracellular parasite CV suppressing this type 1 interferon? Because as I mentioned before, you'd usually see these responses associated with intracellular pathogens or viruses. Although there have been some reported reports in recent years, those immunologists in the room are going, she doesn't read the literature close enough. But it's always been stimulated. There have been reports of a few extracellular bacteria that will suppress this response, but it's always been through the PAM-like receptors. It's always had to go through a more typical path than what is known to regulate epsilon. So I told you at the beginning of the talk that T. vaginalis harbors a bacterial symbiont. There's another one too, but they're both in the genus Mycoplasma. And the one that we work with most, but all our cells have hominis. I don't think we got them from the right part of the world to have the other one. But anyway, it's Mycoplasma hominis. And you can see in the micrograph here of Marlene Benjamin, if you see a nice old-fashioned TM or SEM micrograph of Trichomonas, it's almost always going to be coming from Marlene Benjamin's lab down in Brazil. She did so many really nice early ultrastructural analyses compared to some of the ultrastructures we've seen in this meeting. I say early ultrastructural analysis. But anyway, what she's showing here, this is a Trichomonas cell. And these guys right here are the Mycoplasma. And it's really a very common endosymbiont. This organism has found itself in the female reproductive tract as an opportunist parasite. It's associated with bacterial vaginosis, but it's a really common symbiont of T. vaginalis. It's found in roughly 45% of all clinical isolates, and everything we work with is obviously a clinical isolate. There's no other way to get this bug. And the strain that Josh was working with, in fact, contains this endosymbiont. So he then teamed up with a postdoc in the lab, Emma Betts, who had come to the lab to help us start studying and understanding a bit more the effect of this symbiosis on infection. And so she very kindly decided to work with Josh, and the two of them together decided to ask, is it really Trichomonas that's downregulating the interferon pathway, or is it actually the endosymbiont? And so we start with this culture. That's the one he was working with all the time. So we call it TVMH well, I guess you know what that's for. And you could very easily clear this of all of the bacteria. It's 3 antibiotics and you culture or whatever, 7-10 days, and it's gone. We do our science pretty well. We do the PCR to show that it's really gone and so forth and so on. So then we derive this, TV. And then you can isolate. Emma had figured out ways to isolate the MH separate from the parasite. So we have a separate one that's just the parasite, the bacteria, and then of course we have our mock. They do. The RNA-seq, the PCA plot here just shows you that in fact they separate out quite well. The mock is over here in the green, and one of these this is MH, this is TV. And it's nice, actually. The MHTV really sort of overlaps the two of those. So it was all good, looking pretty good. Oh, wait, I went too fast, sorry. And so then we did an analysis of all the data that came in to determine what was driven by MH only, what was driven by the two pathogens, what was driven by just TV. There's a lot of data here, a lot more work for Josh to follow up on in his own lab now. And there are indeed things that are activated just by TV pathways and vice versa, just by MH, and you'll see all of that if you could read the labeling here. But I want to focus in on the what's called here the alpha interferon response. And what you can see is that it's just activated in the MH only sample. And this shows it maybe in another way a little clearer that activation is purple, not purple, sorry, mustard yellow. And you can see all of these interferon, the pathway genes, response genes, are upregulated here with MH or TVMH, and not not regulated much or even downregulated. And then, of course, as we would have expected, if you treat this with just the TV EVs, you get a very strong downregulation. Now he takes the the reporter gene that came from Sibley, and looks again just to see that, to confirm this data, and he sees what you would predict. You basically get it driven by MH, not by TV. He gets it driven less, about half by TV, the combination of when you have the endosymbiont in the parasite, but he was using, adding equal cell numbers. So that's why it's half here instead of the same as MH. Oh, hey, I think I'm getting close to the end. No, I'm not, sorry. I meant the end of this part. I'll try to speed up. Now I know why so many people said to me, Patricia, please don't talk forever. I mean, well, I could be a little more prepared, I suppose, that I could be able to change the slides and use the pointer. That could have made it faster, I suppose. But yeah, it's, I'm glad you guys said that, because I threw out a lot of data. Okay, I do want to say one thing real quick. I think I won't. Well, I will. Okay, so Josh had another project, and it was a fun project, but I'm only going to show you a very little data here. That was asking whether or not the parasite can internalize its own EVs, okay? And the reason I thought this was interesting and convinced him that he should be interested was that this is from Jane Carlton's lab, the Conrad et al. Paper, that they took a bunch of clinical isolates. A lot came from the ones that they had done their studies on, isolating all over the city of New York, but also from other geographical locations. And they found 11 to 20% of these isolates were mixed. So they had multiple strains. There were one isolate from the same woman, but had multiple strains of T. vaginalis. So it's relevant. Maybe they can talk to each other, and one strain can help the other strain to establish infection. So Josh took a green labeled EVs, put them in with okay, these come from B7RC2 EVs that we've been working with, so very highly adherent parasite, and they really cause, at least for the host cells, an increase in adherence. And this is now back to the wimpy G3 strain, and he fed it to these EVs from that highly adherent strain to this lowly adherent wimpy G3 strain, and then he analyzed to see for uptake. And oh, can I play this? Oh yeah, it's playing. This is with a nano imager, and it's kind of cool. These are actually, he can see, these are the little exosomes floating around in the solution. But what you really should be impressed by is over time, they're accumulating more and more. If I played it again, but you probably saw it. In the beginning, there was a bit of a fluorescence coming out of the two exosomes shown here, but it got, over time, quite high. And that's just showing you the accumulation over time. It's quite rapid, actually. It pretty much peaks even before 10 minutes. So what happens when this is going on? What he was able to show, he and his colleagues, is that the EVs from the highly adherent strain were able to transfer membrane proteins, and particularly this cadherin-like protein that Angelica discovered and studied and showed was an adherence factor when she was in my laboratory. They're on the surface of the highly adherent EVs, and they transfer it over to this wimpy, lowly adherent blue strain here, and then that and some other ligands, Anand's ligand was transferred over. We showed that. I'm not showing you the data. But you'll have to trust me, or you can go read the paper. That resulted in an increase in the parasite adherence of this strain. So even communication between the parasites with these EVs can affect, and does affect, in vitro levels of infection. The last thing, and then I'll move on real quickly, unless you all get up and leave the room. David, David, don't you dare. Oh, it's nice to have old friends, huh? David and I were postdoctoral fellows together. You remember the days? I do. Boy, I was lucky he didn't say any more than that, huh? Okay, the final thing about EVs is now we're looking, and this project is really not ready even to write up, but I wanted to share it with you because I think it's kind of cool. Asking, the host cells make EVs too, right? They make a lot of them, all right? So for a lot of years, I've been wanting someone to do this project. Do the parasites take up the host cell EVs, and does that have an effect on the infection? And Samira Elikay, shown here, which you can't see her, joined the lab not too long ago and said, I'm game to do that. And what you can see here is with the standard uptake assay, yes, the parasites do take up the host cell EVs. But what I think is so cool is if you look at the effect on parasite infection, on attachment, this is when the treated, they're not treated, there's no EVs. And this is what happens to the attachment when the host cells have been introduced. And she incubates them for only 30 minutes. It reduces 5-fold. So it's almost like there's an arms race going on between the host cell and the parasite EVs. The host cell downregulates parasite infection, whereas the host cell EVs, whereas the parasite EVs upregulate infection. So they're trying to see who can outdo the other. I can go through this quickly, I promise. Just to review it. We've shown that the parasite EVs are internalized, and this happens through an EV ligand that binds to GAGs on the host cell. It delivers cargo and promotes infection both by increasing adherence and cytolysis. The EVs themselves dampen the host cell transcription and specifically host cell immune responses, not totally surprising, but we were kind of intrigued by this downregulation of the interferon epsilon. And the epsilon itself is triggered by the common bacterial symbiont, and without the downregulation, otherwise it would be protected against TV infection. So since the EV protects against infection, we are wondering if maybe this is at least part of the story of why a significant reduction in the the interferon epsilon with age may play a role in increased incidence of trichomoniasis in older women. I've just very quickly summarized the whole paper worth of work saying in mixed infections, you get of parasites, you get EV exchange between parasites, including EV proteins to other parasites, and that this enhances the adherence. And the last thing is that the parasites also internalize host cells, which dramatically decreases parasite infection. With this audience, I probably don't need to say this, but I want to say that EVs have been studied now in almost all the parasites, and they play very different roles, but they do seem to play an important role very often in exchanging cargo between the parasite and the host cell, and this affects the actual infection of the parasite. Now you can start walking out. I'm going to try to tell this quickly. I've already told you, I think, that the immune response is very poorly characterized. And one of the reasons for this is that there's no animal models for studying the immune response, at least not yet. People are working on them. And the animal models, particularly the vaginal models that we have that are not even very robust, you have to immunosuppress to get infection. So you don't really want to be studying immune processes when you immunosuppress. But one thing has been known for eons, and that is that neutrophils are recruited to the site of infection. And that's showing you here. This is a pussy. That's basically all that pus is. Well, maybe it's got other stuff, but it's a lot of neutrophils. And this is a cervix that's infected. And of course, neutrophils, in addition to maybe killing the parasite, they're so highly destructive that we think they may be promoting some of this infection-associated pathology. But over all these years, with seeing this sort of neutrophil attraction to the site of infection, no one had ever directly asked, well, are they killing the parasite? And so Francie Mercer joined the lab. She was a card-carrying immunologist, and she decided to approach this question. And she did it by labeling the parasites with cell tracker red, labeling the neutrophils with green CFSE. She co-cultures them. You have to have human serum in here to see anything. She adds human serum. And then she, by fax analysis, by flow cytometry, she looks then in the different channels, red, green, and co-cultured. She looks to see how many survivors she has. So a cell tracker positive, CFSE negative. And of course, I'm telling you this, so they do kill. And this is the killing curve. It's dependent on the number of neutrophils, as it should be. 16 neutrophils to 1 parasite, 8, 4, 2, 1. And what you can see is, we said at 16, it's up to 100% death, and it goes all the way down. But even with 1 to 1, it's killing about 30%. I mean, this is an in vitro assay. Okay, it's not infection, but I mean, it's very robust killing, and it happens very fast. It happens within 10 minutes, and she looked at a bunch of donors to make sure it wasn't donor specific, and I think there's 19 of them here, and more or less it's the same. We do the rest of the experiments at this 4 neutrophils to 1 parasite, because it's in the dynamic range of this curve. And doing those experiments, she wanted to know how this killing was happening. And I'll try to be quick because most people know this. If you go to a textbook and ask, how does neutrophils kill pathogens? This is what you'll see. It's a cartoon. And there's 3 mechanisms that are very well established. One is phagocytosis, where basically just engulfs the pathogen in whole. Another one is degranulation, where the neutrophils shoot out these toxic vesicles that fuse with the parasite and kill it. And another one, which I think is so cool, is it throws out the contents of its nucleus, the DNA, snotty DNA, and it ensnarls this pathogen. And then this is called netosis. This is referred to as forming a net. And then it can also have contributions of your toxic vesicles. So these are the 3. This was published a while ago. I'm going to go, all of this is published, what I'm going to say right now, but I'm going to go quickly here. Francie excluded netosis. Oh, this really moved, huh? She excluded degranulation. This is all published how she did it. So we thought, okay, maybe phagocytosis, that's boring. And in doing these experiments that excluded these 2 mechanisms, she did establish that the killing requires cell-cell contact. Okay, you put a membrane between these guys so they can't touch each other, nothing happens. Okay, so we use the classic inhibitors of phagocytosis, and you know them, cytoclase and warfarin, and sure enough, it would indicate phagocytosis. Here's the vehicles for killing. If you don't have the inhibitors, it kills just like it always has in all those curves I was showing you. You add either, what is this one? I don't know, whichever one it is, one of them, it blocks it almost completely. Same thing here. Okay, so this was consistent with killing by phagocytosis. But as Francie was doing all this work, we had images and we had things that said, we don't think it's really phagocytosis. And I'll remind you that usually phagocytosis will be of a bacteria, like a bacillus, for example, shown here, which is less than 1 micron in diameter. So easy to take up, or easy to engulf something that small. Whereas neutrophils are actually slightly smaller on average than a trichomonas is. And we didn't see a really clear population shift that you'd, in the SPAC plots, that you'd expect if one cell was actually completely engulfing and taking up the other one. And so I am lucky to have known all about, well, I mentored Katie Ralston, who worked in Kent Hill's lab as a PhD student, which is on the same floor as mine. And I convinced her to go to Bill Petrie's lab to do her postdoc because we needed more good people in Anaerobios. And she did. And she had discovered trogocytosis that I think you guys have heard about. Yeah, woohoo! Okay, so that's gonna make it faster. Well, you know, these days, audiences that I speak to, they do start to know it, but it's not that well known still. Okay, so what we did, we were lucky, right? As Francie was getting this data, UCLA finally got its first imaging flow cytometer. And as I think most of you know, it's a really nice method because it allows you to combine quantitation like you get with a flow, but also with high resolution microscopy, and you can look at signals at a single cell level. So here what Francie's done is she's labeled the parasite red, she's labeled the neutrophil green, and then these are the bright fields. So these are the co-cultures, and these are just the individual cells. And so you see here the neutrophil, here the parasite, and here the merge. And you're going to notice immediately. See now, if you tell this story to audiences that haven't heard of trogocytosis, they're like, oh, that's weird. Oh, what's that? You're going to notice immediately that there are what people love to call bites, and the parasite seems to be bitten. And at a lab meeting, she was showing this, and I don't know, maybe it was me, and probably someone else said, yeah, well, I don't know about that, you know. It could phagocytose the parasite, right? And then once it gets inside, it starts breaking it down. Isn't that what they would do? And so we luckily had, I think, a reasonably good way to show this was not the case, because we had learned from a lot of experiments that you can kill these parasites just with 65 degrees heat for 10 minutes. They don't make any ATP, they're dead, but they're intact. They're completely intact. They don't lyse, they're just gone. They're just not making ATP. And when Francie did this experiment and fed the dead intact parasites, as you'll see, you don't get the fragmentation. You just see the intact parasite, and that is, of course, indicating that the fragmentation occurs after death, because otherwise, why did they not fragment up this guy? This is a slide I may not have to tell you guys about. Just in case people in the room don't know, trogocytosis has been around for a long time. It was first described with immune cells that were exchanging membrane and antigen sharing and so forth, but it was not really called trogocytosis. I forgot what it was called. Katie Ralston, who I referred to before, in Bill Petrie's lab, working on Anaerobios, published in 2014 something that was really very exciting for the field, for our entire field, but certainly the Anaerobios field, which is that the Anaerobios takes bites out of the host cell in this cartoon pink, and it keeps taking bites until it compromises the membrane enough that it kills the host cell. And so, yeah, I think you had a talk on this. I think I was on the plane or something, so trogocytosis is now established as a mechanism by which the parasite Anaerobios will kill host cells. There was a Dutch group, and they're still doing nice work over there. There are also in 2016, not a Dutch group, this guy's up at Berkeley, I think, that was showing that intracellular bacteria can be exchanged between macrophages using this process of trogocytosis. And it's kind of cool because that way, you know, the bacteria could spread through macrophages just, you know, by the macrophages biting each other. And the Dutch group showed that neutrophils kill cancer cells by trogocytosis, literally as we were writing up this paper. So finally, there was first report of a neutrophil doing trogocytosis. So Francie wanted to know absolutely for sure if trogocytosis was happening, was preceding death. So she did this assay where now what she's doing is she's labeling, limited labeling on the membrane of the parasite, which is green here. She's not labeling the neutrophils. You just see them in the bright field here as dark gray things. And she throws in propydium iodide too, because what happens with propydium iodide is that it won't enter the cell. It stains DNA, the nucleus, but it won't enter the cell unless there's the cell is dead. And what you can see is you're getting all this uptake of bites of the parasite into the neutrophil, and then only much later do you see the nuclei stained with propydium iodide. So the killing occurs before death. So the trogocytosis is the killing mechanism. She showed the average number of PMNs. Oh, sorry, that's neutrophils, but another name. For this, it was 3 to 6. The number of bites before death, 3 to 8. And the time before from first bite to death on average only 8 minutes. So it's a very fast process. Okay, I just love showing this. I'm gonna skip most of it. You guys have seen much more fancy and beautiful microscopy this week, but this is just showing 3D super resolution with, I just took a slice, I didn't bring a movie, with the bites in. The reason I love this picture is because, and Francie sees this a lot, you see it over there too. What you see is that there's a lot of neutrophils. Oh, they're magenta, and this is the parasite. You see a lot of neutrophils all swarming at the same parasite, okay? But what I think is so cool is in many, many pictures, only a few of them are doing the trogocytosis. And we see this repeatedly. We haven't quantified it. We really need to, or somebody needs to. I'm just closing the lab. But it's like they're a team. They're playing a team sport, and they all swarm in, and these guys are holding the parasite in place because this is it's got 5 flagella. I told you, it can swim, but only a few bite. Cool, huh? Is that not cool? You like cooperation? And this is the part Francie told me I'm not allowed to say, is it looks like the biting ones are near the flagella. Wonder what that says for how they decide where and how to signal. Anyway, I'm going to really quickly tell you that serine proteases of the neutrophil are involved, and I'm not going to go through the data. I think you're probably happy for that. Okay, so I've shown you and told you that the neutrophils are able to kill by trogocytosis, and that this is very rapid. They start seeing killing in 2 minutes. I mean, this is a rapid process. Okay, but what I haven't told you is that we've looked at, I'm only showing you 4 here, but we've looked at 9 different clinical isolates of T. vaginalis. The one that Francie was working with for her study is G3, which is very highly killed in that very rapid assay. And there's another one we isolated ourselves in LA1, also very highly killed. But here, look at these 2. At 16 to 1 and 8 to 1 ratio of neutrophils to parasite, not very well killed, okay? And we've looked at about 9 strains now, and about half of them are killed by trogocytosis, about half of them are not. And so at this point, Katie Moritore, who I'm going to say is Diane's cousin, or second cousin, who is Diane's second cousin, and she may be, maybe not quite as bright as Diane, but she joined my lab. I was so happy to have her. then COVID came and she was like, I can't stand this. I can't work in the lab. I'm bored. And she went to do, high level analytical chemistry at Amgen or whatever. She's an analytical chemist. I kept trying to teach Katie to do a sloppy experiment. Oh my God, that doesn't happen. But she joined up the lab at that point. She was also a technician with Jay Baggs, but I guess Jay didn't come here, but in case he's listening, hi, Jay. And hi, Katie. She joined up with Francie and decided to ask what differs between the parasite strains, the ones that are rapidly killed as opposed to the ones that aren't. And if you're just a geeky cell biologist, that's more interesting than the killing itself. What's different? And we came up with a bunch of hypotheses. I don't think I'll even go through them here. You can read them. But the data's been reported finally just this year in bio. But these different hypotheses we had that would cause some strains to be resistant to killing, we eliminated a number of them. The first three is shown here. The data are published. But my favorite one, and I thought it was going to be the reason, was that the resistant strains just simply prevented neutrophils from swarming, like here, this is a swarm, and biting. If they don't swarm, they're not able to bite. That prevents them from trogocytosis. So Katie and Francie looked at a whole bunch of these. You can count the numbers, the dots. And what they looked at was for swarming. These are the two trogocytosis sensitive strains. Okay, they're swarmed pretty well, aren't they? But so are the trogocytosis resistant strains. Okay. Same thing for biting, sensitive resistance. So they swarm, the neutrophils are attracted to them. They swarm the parasite. They even bite the parasite, but they don't kill the parasite. And so our last hypothesis that we tested, Katie had gone to Amgen. Sandeep Mukherjee did these experiments, was that the resistant strains are better at membrane repair. And we got a hint of this from the Dutch group again that do these nice studies of trogocytosis of cancer cells, where they were looking at cancer cells that would undergo change so that they were sensitive, but they would become resistant to trogocytosis. And they found that it was a calcium-dependent vesicle tethering fusion type of mechanism that ended up with repairing the membrane. And so Sandeep takes our two sensitive strains, our two resistant strains. He incubates them in the presence or the absence of calcium, the permissive or the restrictive temperature. Then he adds listeriolysine, so a well-known whole pore-forming protein. This is just a standard assay that's used a lot. He puts the listeriolysine in the presence of a zombie fluorescent dye, and this zombie dye will not enter the cell unless the membrane is compromised, unless they often use it for looking at dead cells, but even compromised membranes will allow the zombie to go in. And so the question was, do the resistant parasites reseal their membrane and let less dye in? And since I'm telling you this, you're going to know the answer is yes. So the y-axis are different values, so it's confusing. It looks like you should be able to compare them directly by eye, but you can't. But what you see is here, this is like adding the listeriolysine. I'm sorry, I'm so sorry. This is adding the zombie dye, but without the listeriolysine. So this is your baseline. So all their membranes are pretty darn intact. They look pretty good. And that's true under both conditions. But when you have the gray bars, which is adding in the lysine and letting the dye be taken up, what you can see is that much less dye is taken up under the permissive conditions, which is what we would have expected. And the resistant strains take up much less dye than than the sensitive strains, which is consistent with them doing a very much better job of resealing those membranes. But this led us to another point, which is if trogocytosis sensitive strains are killed, much better than trogocytosis or even trogocytosis resistant strains not being killed, over evolutionary time, you shouldn't have trogocytosis sensitive strains, right? It doesn't work that way. Or you should certainly have a whole lot less of them. Now, we haven't done a really thorough look at all the different strains out there. But like I told you, what we've seen so far, which is only an N of 9, which is nothing, it's more or less 50-50. I've also told you the trogocytosis killing is very rapid. And those of you who know neutrophil literature and work with neutrophils or are familiar with neutrophils know that netosis, on the other hand, is very delayed. It's sort of like a last ditch to get the pathogen. Can't get it any other way. Okay, we're going to throw we're going to commit suicide, throw our nets out, because they're committing suicide. But neutrophils are good at that. They throw their nets out and capture the pathogen. And this happens much later, many hours after confronting pathogen. So we looked to ask if you have a longer incubation with these trogocytosis resistant cells, will they actually be killed, and will they be killed by netosis? And as you can see here, here's the killing of those strains at 1 hour, and here's the parasite death at 16 hours. So much more so. And if you add DNase in, which is sort of the standard way that people start their experiments, at least to see if it's netosis, you see that now the parasite death is knocked down substantially. It's significant, but it's okay. So this argues that what the trogocytosis resistant strains are in fact killed, but only later in time, and by the mechanism of netosis, which establishes that neutrophils use different strain-specific mechanisms to kill. So I'm going to summarize this real quickly, and then we'll be done. Trogocytosis was identified as a mechanism that neutrophils use to kill pathogens, which had never been shown before. Francie in her own lab has shown that this happens also with T. fetus, which is not terribly surprising, but at least that. And I think has plans to try to look at other really large, sort of highly motile pathogens to see if maybe it could be happening with them too. The killing, trogocytosis killing, is strictly contact dependent. It happens very rapidly. The bites are taken up prior to the death, so the trogocytosis itself is doing the killing. Not all T-batch strains are killed by neutrophils, and those that are resistant to the rapid killing seem to be better at repairing their plasma membrane. But hey, they may be better at doing that, but ultimately they get done in by netosis from the neutrophils. So there's different mechanisms. And this, I think I've acknowledged, a lot of these people as I gave the talk. But in addition to them, who are listed all here with their pictures, there's been a lot of other contributing lab members over the years. In fact, I'm sure I left some people out. My memory's not what it used to be. And I want to also acknowledge my only funding source, National Institutes of Health, until 7 weeks ago, when they suspended all of my funding. So thank you. But I think, okay, I don't want to get political. I'm surprised you didn't talk about how damn political I am. I don't want to get political, but NIH would not have done this. His initials are DJT. And it was a dictate that had to come down. But what I told you was in fact funded by our federal government. And thank you so much for your attention. I hope I didn't go over too terribly long. David, am I all right? I even got the okay from David. All right. And so I'm happy now to take any questions. Thank you.
Thank you very much. That was fantastic. It was fantastic to see all these hypothesis-driven experiments. Marvelous. I have time for questions. We have one microphone, so bear with us.
Oh, you can have this one back. I can stand here. Here. I can stand at the.
That's great. So really quick, I was just wondering if the host EVs are protecting from infection or attachment, do the host cells generate more EVs after they're exposed to trichomonas? Does that, or like, are the cells always?
Good, very good question, but I don't know the answer.
okay.
I do not know the answer. The way we work with the host cells whenever we're trying to isolate EVs, we intentionally, of course, don't have the parasites around because then we would have a mixed EV. But we could probably do that if we could differentially label them, but we haven't done it. We don't know. But it would be a clever thing for them to do. It would be a good strategy, but we haven't looked at it, no.
Okay, hello, I'm Fifi from UGA. Great talk. I wanted to ask.
Oh, hi.
Yeah, I wanted to ask if the parasites are able to, let's say, communicate or transfer EVs from one to the other. Can the resistant ones also transfer EVs that have some resistance to make the ones that are susceptible also resistant?
Oh, good question. I mean, you've read about the malaria EV projects, haven't you? You know, I don't know the answer, okay? Give me a second, I might know the answer. I mean, I have to think about, we don't know the answer because we haven't looked at resistance, but the strain that I was showing you is, it's not great, incredibly highly resistant to metronidazole, but it's more resistant to metronidazole than G3. So I could do that experiment, I haven't. We have no reason really to think that EVs are playing a role in resistance. I think, at least in the malaria world, correct me please if I'm wrong, they were basically, they were transferring resistance of factors that had been introduced into the EV, right? They don't play a, is there an evidence, I'm sorry, I'm not supposed to probably ask you guys questions, but is there evidence for EV to play a role in drug resistance in malaria that I'm not aware of? Okay. But we could do that experiment. I mean, this is the thing. I don't want to shut you down because the number of times that people have said, is it this way? Could that happen? And I'm like, yeah, they'll tell you. And I'm like, come on, that won't happen. And then they go to the experiment, it does happen. So this is how you learn new things.
Hi, I was wondering if you thought that the bacterial endosymbionts had any effect on the trogocytosis resistant versus not susceptible lines?
So whether or not you do or don't have the endosymbiont.
If that has any effect on if the trichomonas can be trogocytosed or not.
Oh, if it can be trogocytosed or not. I see what you're saying. We haven't looked at that. Oh, yes, we have. Yes, we have. Yes, we have. I'm so sorry. Yeah. There was a miscommunication between Francie when she left the lab to set up her own lab and the new coming postdoc that took over some of these Sandeep. And Francie always cleaned out all the M. hominis. I don't know why. She just didn't want the M. hominis around. And she'd do a PCR before his experiment. She always knew they were super clean, okay? And Sandeep and she kind of miscommunicated. And he didn't. He was working with G3 that had hominis, and their data matched beautifully. In fact, surprisingly well for, turning a project from one person to the other. Yeah, so no, there is no effect.
Hi, Patricia.
Hi.
Hi. So my question deals with the LLO treatment that you did, and seeing the differences in ability to repair membranes that you saw with listrolysine O.
Okay.
Yeah. So one possibility could be that doesn't have enough cholesterol on the membrane. So you don't get the LLO to work, the accessible cholesterol. So difference is maybe because of that rather than ability to repair the membrane.
Or that with your repair is better with more cholesterol.
Yeah, so if you have more cholesterol, you'll be able to get the whole.
Yeah. So it's not the fusion of vesicles per se.
Right.
Okay, the one thing that you could be right, but the one piece of data that's published, but I didn't show it today, is the trogocytosis strains are much more granular. When you look at them, they have granular, meaning they look like they have a lot more vesicles.
Yep.
But I hear what you're saying.
Yeah,
And you know, we had to hurry up to write this. Get it out. You can't hear me now, I guess. But because, and I was shaking in I was shaking in my boots that the reviewers were going to go this. Give us more on the repair. Because we don't really know how it's working. Yeah, we have a hypothesis.
Yeah. Hi, Zachary from the Whitehead. I was curious, do you know if there's a series of like diversity in EVs that are released by this parasite, like other small EVs or even large EVs?
Oh yeah, Natalia here can tell you all about it. There's a range of vesicles that are herself in her own lab has much larger group, larger in diameter. They're called exosomes, and they seem to have some of the same properties, different properties, but also some of the same. So there's a range, yes, and range of different vesicles that can be released. And in fact, you know, Randy Sheckman is really hardcore that to be an exosome, you have to express CD46, I don't know, but there's markers on mammalian exosomes because there are so many different types of vesicles and different sizes that maybe are really doing different things. But we don't have that type of marker. Those are all mammalian markers. But they do.
So Patricia, over here, I have what's almost certainly a rhetorical question, but I'll ask it anyway. As you said, we've known each other for a long time. We've probably each fallen asleep in many of each other's talks. That's not the question.
Oh, that wasn't the question.
The question. So we've known each other since we were postdocs together. As you've heard from Natalia, most of us were pretty intimidated by Patricia's spectacular success as a postdoc, and yet in moving to set up her own program, she moved to work on Trichomonas. And we've heard from Natalia what a big hole is left in the Trichomonas field with shutting down your lab. So with all of the junior investigators in this audience, my question to you is there room for more research in Trichomonas?
There's so much room for more research. I mean, I think that's how Josh, my last postdoc that just left to set his own lab, I think that's how I was able to convince him, you know, to come, because I actually said, I'm just closing this lab down fairly quickly and you can have everything. And so there's a lot, but that hasn't already been taken by other people. But there's a lot of room to fill. And it's not a difficult parasite to do biochemistry on. You've seen it. What have I shown you? I've mostly shown you biochemistry. I've shown you some cell biology. It needs more people in the field because we need people who will really work hard to get the molecular biology, the gene knockout, the gene knockdown, all those sort of really powerful genetic tools better established. So yes, please. Contact them to go.
You might want to use the microphone because there's another audience online.
Oh, well, they didn't really.
And that's actually a good transition to a question online, and will probably be the final question because people have to run to dinner. I think, so this is from Jane Carlton.
Oh, hi, Jane.
So there were actually 50 people online watching you. So,
Watching me make a fool of myself?
No, not at all, not at all.
A distinguished professor who can't use a pointer?
So Jane is asking, what words of wisdom can you impart to the next generation of researchers in the current NIH funding climate for infectious diseases and especially women's health?
Ooh. Well, I don't know about the women's health part. Hi, Jane. I don't know about the women's health part so much. But you know what I have always told my trainees is that you cannot control the funding levels. You've heard this many times, both of you. You cannot control the funding levels. There's so much that you cannot control. But if you have a passion for what you're doing, what you can control is how well you do it, how much you read, how much you think, how well your experiments are controlled, et cetera, and just, get in there if you really have a passion to work in this field or any field in experimental science. Get in there, do your best, do what you can control, and see what happens. I can't be a lot more optimistic than that because there is a problem right now, and we all know that in this nation at least. But if you don't want to do parasitology in America, they've got some damn good labs in the UK and in France and Germany, at Switzerland. That was, Jane, I know that wasn't the right answer.
Okay, all that's left then is let's thank Patricia again for a fantastic talk. Thank you.
Thank you very much.